Hello again. This is the additional video from the last one, which is about animal and plant cells. Uh, this is not for your revision for say because this would not be in your exams, but this is just for additional information and know what you're gonna study in the future if you study biology. And there's a part about mitosis, which we'll get into that. Yeah. So the last video, there was a very basic diagram of a animal cell, which only contains like the cell membrane, the nucleus, mitochondria, and such. But now this is a complex version of an animal cell, which contains all the organelles, and which contribute to what the cell does and how it generates like, energy and stuff. Uh, and now we have the plant cell, which, like last time, contains like the chloroplast, the plasma membrane, which is the cell membrane, the cell wall, and the nucleus. As you can see, there are also the endoplasmic reticulum and Golgi complex or apparatus, which are present in both plant and animal cells. I'll cover that particular organelle because it's easy to understand and really like present in both, so why not? Right. So here are the two organelles which I would cover today, which is the endoplasmic reticulum and the Golgi apparatus. So the endoplasmic reticulum has can be either smooth or rough. Rough would be studded with ribosomes. So what does the ER do? It's generally transporting materials between organelles. So the smooth ER transports lipids and the rough ER transports proteins. You can look into it yourself if you want to. So the Golgi body, the Golgi apparatus, or the Golgi complex, which are the same thing, has a structure of like folded membranes and looks like the Spotify sign. So yeah, what does it do? It is involved in sorting, storing, modification, and export of secretory products. So if you want to look into it, you can learn how the ER and the Golgi apparatus work together to transport materials. Yeah. So now we move on to the additional microscope information. So last video I covered the light microscope. This time I'm going to cover the electron microscope. So light microscopes could be found like in your laboratory or you can buy it, but electron microscopes are very large and could mostly be seen in like universities or used in universities. So the electron microscope uses electromagnets instead of lenses, which are found in like the light microscopes, to focus electrons, resulting in a larger magnification and resolution. So these photos on the left are viewed under a electron microscope. So as you can see, they are black and white because an electron microscope only can view black and white and dead specimens, because if it's moving around you can focus on like the electron. So now we cover on cell division. This is a really complicated procedure which most of you might not understand. It took me a while to understand first. It's really complicated, so I'm just going to simplify it to the max. So first stage of the cell division is the interphase, which the cell grows and DNA is replicated, okay, and prepares for mitosis and cytokinesis, which are the later stages. So basically the interphase is how when the cell grows and DNA replicate. Okay, I'll cover mitosis on the next slide, but just remember this. And now we have cytokinesis, which means that the contents in the cell are segregated and the cell splits into two. So let's move on to mitosis. So this is mitosis. Uh, so you can just look at the picture to see if you understand, but mostly you don't understand. So what happens in the prophase? So these things that contain genetic materials called chromosomes, they first condense and the nuclear membrane dissolves. Okay. Then on the next stage, metaphase, these things, the chromosomes, align in the middle of the cell because of these things called spindle fibers connecting to it, which are produced in the centrosomes. Okay, this is... Now, on the next stage, there's anaphase, which the chromosomes split into halves and move to opposite poles, as you can see in the picture. And the last stage, telophase, is when the nuclear membrane reforms, they decondense, and then cytokinesis, which is covered in the last slide, they split it into two, so then two identical daughter cells would be formed. Mitosis is a really complicated procedure, and it is hard for lower form students to understand, but if you get the idea of what is happening here, this would aid you in the future for understanding like mitosis, meiosis, and such about cell division. I find it very interesting on how this separates. So if you find it interesting, you could study biology. Haha. So I'm yeah, I'm not gonna mention meiosis. So yeah, hope this makes biology interesting for you. So have a great day and goodbye.